Hello, and welcome to ACR Journals on Air podcast. I am your host, Vicky Shamagam. Primary Sjogren syndrome is a common autoimmune disease in which there is chronic inflammation of exocrine glands, particularly those producing secretions such as the salivary glands and the tear ducts, with the disease manifesting as sicker symptoms of dry eyes and dry mouth, often accompanied with profound fatigue and sometimes other autoimmune manifestations. The manuscript we will discuss today is Association of Circulating Antibody-Secreting Cell Maturity with Disease Features in Primary Sjogren Syndrome. It was recently published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. I'm happy to be joined by the manuscript's first author, Dr. Tobit Schijnmets. Dr. Schijnmets is a postdoctoral research fellow at the University of Groningen in the Netherlands. He completed his master's and PhD at the University Clinic in Erlangen in Germany, including a research internship in the lab of Jason Sister at UCSF in California. And he is now building a research career in Dr. Franz Kruz's research group, studying B-cell function in Sjogren syndrome. Welcome to the show, Tobit.
Thank you, Vicky. I'm very excited to be here.
So as you know, many of our listeners are clinicians looking after patients with Sjogren's, and some are also patients with Sjogren's. And we know one of the major manifestations of Sjogren's is autoantibody formation. And some patients also have lymphoepithelial lesions, or LELs, where the B cells infiltrate into glands, contributing to glandular dysfunction. Share with us, why did you decide to study B-cell maturity in Sjogren's?
So at first, B cells are very prominent in Sjogren's disease. So many people have elevated levels of total IgG antibodies in their blood. Also, a lot of patients have autoantibodies that are targeting their own tissues. And the frequency of different B cell subsets that can also be quite altered in the blood of the patients. So for example, you can have less B cells that have memory functions and more B cells that are very fresh and naive in their development. And if you look at typically affected tissues, like you already named the salivary and the lacrimal glands, the B cells are also very present there. And they can even form germinal center structures where B cells basically get trained and can also form then antibody secreting cells. And also some parts of the salivary glands in some patients can form these lymphoepithelial lesions that you named. And B cells can infiltrate those structures and reshape the salivary ducts in those structures.
And so you wanted to look at these antibody-secreting cells specifically, or ASCs, and particularly to focus on markers of maturity in those cells. And you used a technique called high-dimensional spectral flow cytometry. So firstly, can you share with us what high-dimensional spectral flow cytometry is, and how does it differ from regular flow cytometry?
Yeah, so in general, flow cytometry is something that, most immunologists are doing on a very frequent basis. So you can read out different properties of cells by using different colors that you are linking to the cells, and that you can read out in a machine for, is the color present, then is also the property of the cell is present, and you can also read that out in a quantitative way. And the spectral flow cytometry is just a more advanced method that is also allows that you can acquire more colors than with the conventional flow cytometry. So it can, for example, with conventional techniques, it would be difficult to separate green from a yellow-green color. But with the spectrum, you can distinguish now those colors, and that gives you a much bigger panel of colors that you can use for your analysis.
And so you're really looking at more cellular markers in that panel. Is that fair to say?
Yes. So basically, every color is like a dimension then. And the more colors you use, the more properties you want to analyze for your cells, the more dimension you add to your data.
That makes sense. So tell us a little bit about the patients you selected for this study. Who were the patients? How did you enroll them? And who were the controls?
Yeah, sure. So here in Groningen, we have the very big advantage that we have a very big study cohort of Sjogren's patients because we are a Euro Center of Excellence. So we get referred a lot of patients from all over the Netherlands to our physicians that they can diagnose Sjogren's and also treat the patients. And yeah, as I said, we have a longitudinal cohort with a lot of patients that also provide samples for our biobank. And that includes blood samples where we can isolate PBMCs from, and that formed the basis for the study. But this biobank is also linked to a database where we can store the disease parameters of the patients.
And who were the controls?
The controls were blood samples from healthy individual that we selected that they would match the age and also the female-male ratio of the patients.
Okay. And so were the samples collected in real time or were they frozen in the biobank? And does the storage of the PBMCs impact ability to accurately define the cell markers of maturity that you wanted to study?
Yeah, it would be better to use actually fresh samples for a couple of reasons, but logistically, that is much more difficult. And we use only frozen PBMCs for our study, and that can have the negative impacts that antibody secreting cells, they don't really like to be frozen or thawed again. So some of them might just die before you can analyze them. And there are also some indications from other papers that some markers you want to read out with the flow cytometry are a little bit harmed by that whole freezing process.
Although I guess it does allow you to do the flow cytometry in batches, which gives you some consistency in that process.
Yes, correctly. So we chose also to use the frozen samples because then you can do as many samples as possible within the same experiment. And that also reduces your batch effect.
So that impacts variability, I guess, in your results?
Yes, that can have a big impact on actually the quantity of the markers that you want to read out. And this were quite a big part of this study to read out quantities.
Okay. And so in the flow cytometry portion of the analysis, you have some really beautiful figures where you analyze the groups based on immunoglobulin levels and the presence and absence of lymphoepithelial lesions in the patients. Can you talk us through what that analysis showed?
Yeah, so in general, Sjogren's patients have elevated levels of IgG antibodies in their blood. So normal range would be 6 to 12 grams per liter. And our patients had about 20. And one of our main hypotheses was that if the patients have more antibodies, that should also be reflected by either more antibody secreting cells or they should be more active, that they secrete more on a cell to cell basis. And we also expect that this would be reflected in the numbers then of the cells or in their phenotype. So we made this two groups with Sjogren's patients that have high levels of IgG in their blood and the other group that has lower levels of IgG, and wanted to compare those different subgroups on the antibody secreting cell level. But yeah, to our disappointment, we didn't find much what is separating those groups. Definitely not frequency-wise. If you can, as you can see in the paper, there's no difference for those two subgroups. So we looked for other factors that might explain different frequencies of ALCs in the blood of the patients. And the small indication came when we looked at LAL positive and negative patients. And actually, there we saw the biggest difference between those groups if you look at the frequency of the antibody secreting cells.
Interesting. And you also completed a maturity index for the antibody secreting cells. Tell us about how that maturity index was developed and what did you find in that portion of the analysis?
Yeah, this is a little bit longer story, if I may. Yeah, in the field of plasma cell scientist, it's rather small, I would say. But most people would say if a plasma cell or an antibody secreting cell is not expressing CD19 anymore, this is a mature cell.
Mm-hmm.
But in recent years, there has also been some studies published that look at CD19 plus and minus AACs. And for example, they compared them for the specificity to the Vaccinia virus, which is basically a readout for smallpox.
Mm-hmm.
And smallpox was, as we know, eradicated in the 80s already, before I was even born. And these, in those papers, they could still find antibody secreting cells that were expressing CD19, but were specific for this smallpox virus. So the cells must have stayed in the people's body for quite a long time, actually. And that gave us the idea that the CD19 plus minus paradigm or hypothesis is less strict than most people assume. And actually, there are already a couple of other markers known or described that are also changed with the maturity of the AACs. And these can, for example, be CD28 or also the HLA-DR.
So you're experiencing the evolution of the science.
Yes. And our idea was then to try to combine multiple markers that are already previously shown to be linked to the maturity of the antibody secreting cells to get a more reliable readout. That the more markers you have, the more it should reflect the real maturity of the cell. That was the idea behind that. And also, actually, we found when we calculated this maturity index, that is combining 4 different markers that are already suspected to change with the maturity of the cell, then we could see in some Sjogren's patients they express very high levels of CD19. So the cells on the first glance look quite immature. But if you look at CD28 and CD138, they're also quite high for those markers. So on the second glance, they might be much more mature than you think first, and they might be overlooked with a very simple only CD19 analysis.
And just to clarify for our listeners, so that's not seen in the control patients?
In the control patients, you also see that to some extent, but the maturity in total was a little bit lower in the controls than in the Sjogren's patients.
And you did an analysis that was something I've never heard of, so I'm hoping you can explain it to us, called T-distributed stochastic neighbor embedding. Can you tell us what that is? The plots that you've generated look a little bit like turtles, but that's really just a description. Can you help us understand how we interpret them?
Yeah, I thought that is quite funny because I never thought about turtles when I looked on those plots. Usually it also looks more like clouds, but let's start from the beginning. So most people just call that TISNE as the abbreviation for the method.
Mm-hmm.
And it's a mathematical formula that is reducing dimensions. So as you think back to the questions we had earlier about the high dimension spectral flow cytometry, this is coming back to the same problem that if you want to display more than 2 dimensions, you can't do it on a paper anymore. And if you have 4, 5, 6 dimensions, humans can't really grasp that with their mind. So what this computational approach actually does, it looks for all those 20 colors, those 20 dimensions that we had in our analysis, and tries to find for each cell to cell how alike are these with the background of all these 20 dimensions. And the more alike 2 cells are, the more closer they get on these t-SNE plots. And that also means usually you would, for example, if you use that in a whole blood sample, then you have a whole bunch of very different cell types in there, T cells, B cells, monocytes, erythrocytes even maybe, and they would be on very different sides of these t-SNE plots with actually quite a lot of white background between them because they separately quite nicely from each other. But because our analysis here is just antibody secreting cells, they already have quite a similar phenotype. And that's also why they are forming that more Turkle-like structure and not like clouds. Or you can also think about a group of islands, for example, how it usually looks like.
So I'm trying to think of a metaphor. You're essentially using the math to analyze in 20 dimensions. So it's like we've kind of gone into galaxies of outer space to understand how these cells are related to each other.
Yes, you can do that. For us, it was quite a simple, or it's not that simple, but it can visualize quite nice the properties of the cells all combined.
Mm-hmm.
And also, usually, it is a quite unbiased approach that you run this t-SNE analysis and then you look at it. And then if there are main differences in your cells, that should be visualized quite well. So, for example, we saw that one group of cells was really highly expressing CD138. And then we also looked, where do these cells come from? Do they come from the controls or from the patients? And it turned out that almost all of these cells came from the patients. So then you can make the conclusion that cells from the patient express more of this CD138 marker.
And using that maturity index, you then correlated the maturity index data with clinical data, including the EULAR Sjogren's syndrome disease activity index, the ANA titer, the focus score, and ocular staining scores. Can you sort of put that all together? So that was adding an additional dimension, the clinical dimension. What did that analysis show?
Yeah, sure, Vicky. I can try at least. So from also the t-SNE, we saw that the more AACs that were coming from the patients, there were also the tendency of these groups of cells to express the receptor CXCR3. And that usually is referred to cells that were generated in a pro-inflammatory context. So we had some indications that we then also saw on a patient level, that patients that had higher maturity in their ACs, those cells also expressed higher amounts of these CXCR3 receptors. So that gave us an indication that there might be a link between the maturity of the cells and their inflammatory phenotype.
Mm-hmm.
And then from that, we went on and asked ourselves, is that also reflected by these variety of disease parameters that you have in Sjogren syndrome? And for example, the SDI you named first is a global disease activity index that should reflect a lot of different aspects that the patients are suffering from, but then also, of course, more detailed parameters like the focus score reflects how many immune cell infiltrates the patients have in their salivary gland. And there we also saw correlations between the maturity of the AACs and then, for example, the focus score, or also the titers of anti-nuclear antibodies in the blood of the patients.
And so I'm interested sort of how you pull this all together. Do you see this having implications for kind of using these technologies to either improve diagnosis or possibly as a predictive tool for prognosis for patients with Sjogren's? Is your lab kind of pushing forward in that area?
Yes, definitely. So that is our plan. We were not really able with that paper yet to nail everything down. But our idea is that we first can have this maturity index as some kind of biomarker that, for example, you can identify patients that have higher disease activity, or that it might also mark special patients that could benefit from different treatments that are now used for Sjogren's patients. Because we also started with there are a lot of B cell abnormalities in the patients, and actually a lot of studies in the disease were already trying to deplete the B cells in Sjogren's disease, but this had not really good effects. It was not convincing so far. So the idea of our group is also that the antibody secreting cells, they are coming from the B cells, but they look quite different on a cellular level, and they would need different treatment to deplete them. And probably our work can give some indications how these antibody secreting cells look in the disease, but also potentially give implications for the future, what kind of targets on antibody secreting cells might be there that are interesting for Sjogren's.
Yeah, and I think it's really interesting kind of clinically, because as clinicians, we know there are some patients with Sjogren's who really do totally fine. They, have fatigue and obviously the sicker symptoms, but really not super aggressive disease. And then there are others where the disease is extremely inflammatory and aggressive and, can be life-threatening. And so if this is in some way helpful for subcategorizing those groups, it would be really helpful from a clinician standpoint. And I want to make sure, am I understanding correctly, that you needed this high, the spectral flow cytometry to be able to develop the maturity index, but now that you've identified those specific markers, this could be something that could be used on a more basic flow cytometry type scale?
In principle, yes. So at the moment, we also have follow-up projects that are trying to refine that method a little bit better. But the idea would be that we come up with a core set of markers, might it be 4 to 6 probably, that a lot of people can run in their own labs that don't have a spectral flow cytometer in their facilities, who are restricted to just being able to read out 10 colors, for example, or 12 colors, 15 colors, that they can also apply that method. And that would be the idea in the end, to give some kind of guidance and say, yeah, you don't need all of these markers to really describe the cells properly. Yeah, that would be the final stage for
and then follow-up question to that. These are markers that are relatively stable on these cells over time. So in terms of sort of this would be a one-off test you would do for prognosis, or is it something that would need to be done at different time points of disease, or you're really not at that stage of being able to understand the process yet?
So at the moment, our working hypothesis is that this is something also disease specific, that if you look at other diseases, are they rheumatic or autoimmune, that it might be different how the AACs might look like according to their maturity. It might also be lower, for example. In a very related disease in lupus, there are a lot of indications that antibody secreting cells actually might be less mature than in healthy people. One of our working hypotheses is that it is disease specific, but we also have some very fresh data that it might be very stable in individuals over time.
Good. So that will be helpful clinically, I feel like, and will allow this to sort of scale more easily into a clinical setting. So that's really really
if
exciting.
it changes on a weekly basis, then of course it's not a good readout parameter.
Yeah. It just becomes much harder to kind of interpret from a clinical standpoint. Oh, Tobit, so thank you very much. That was extremely helpful. And I really appreciate you distilling that so that we could all understand and get up to speed on your science. I wanted to change tack now and discuss your career. You have had quite an exciting career pathway so far. You've lived and worked all over the world. And I'm wondering if you would be willing to share with our listeners your experience as an international scientist. Are all research labs essentially united by that common language of science, or do you have tips for our listeners who might be interested in moving internationally as scientists? How did you navigate the international transitions?
I think first I would say, if you have the opportunity to just go somewhere else and you don't have really a very good reason why to stay in the place where you currently are, if you finish the project, if you are ready to start something new, you should really try to find a different location to also do the research you like. It takes some courage, of course, but usually nobody will regret it afterwards. But you also refer a little bit how research is done in different places.
Yeah. So tell us, I think, as an international scientist, and I guess you did a elective over in California. How did you find that experience? Are all research labs kind of talking the same language, or did you have to sort of learn to translate a little bit your science to fit the different cultures of the different labs that you were entering?
So my feeling was that if you talk about the science, it's basically all the same. There, it
Mm-hmm.
doesn't really matter where you are. If you interact with people, of course, then different backgrounds give different interpretation of what you're saying, how you're behaving, and this might take some time to adapt, but this is always doable.
Yeah. Yeah. And sometimes it's energizing, right?
Yes, it gives a completely different point of view, and that, of course, also opens your mind to different possibilities. And I think you just tend to think broader in some
Mm-hmm.
ways.
Yeah, and super important for science, I feel like, to have that diversity of thought. And I think it's one of the things that allows scientific labs to kind of push the boundaries of the research.
Yes, but as a master student, I was quite surprised how day-to-day lab practices were actually quite similar. And
Yeah.
it doesn't really matter if you do it in Germany or in the Netherlands or in the US. A pipette is still a pipette, and if you have the reagents, you can do the nice research wherever you are.
Yeah, and I think it's important to sort of remember that, as particularly early in your career, is that being brave enough to have those different experiences is important and gives you a different, I guess I'll use the word you used earlier, a different dimension to your career growth as well, right?
Yes, definitely.
So you are now a postdoc in Franz Kruz's research group. Tell us what attracted you to that lab and particularly that area of research. Do you have any tips for others who are trying to find a postdoc position, and how did you assess opportunities and trying to find a supportive lab and environment to work in?
Yeah, this is also quite a story, I would say, or there's a little bit background around this. So I did my PhD already on plasma cells or on antibody secreting cells, but in the context of mouse studies, and it was a very basic project. So I generated knockout mice and analyzed the particular function of a protein and what happens to the B cell compartment in the mouse if this protein is missing. And there was not at all any disease context applied. It was, very basic research questions in mind. And this work, I say, is very important. And if nobody's doing it, then there's also no background knowledge that other people can use. But after around 4 years or so, I also tended to not really want to work with mice anymore. So I had the wish that I shift a little bit to translational research, but I also wanted to have my work to have more impact, that it is closer to really give benefit to patients. But I wanted to stay with the antibody secreting cells. So that might also be a tip for your question earlier, that I try to change enough that you learn new things, that you are challenged, but you should try to keep a little bit familiar, that it's not too hard to get adapted in a new environment. And I try to keep to this scheme. And actually, my supervisor now, I met him at a conference. I went to a conference about B cells some couple of years ago. And actually, I didn't know anybody at that conference. So my colleagues didn't go there. And I just started to talk to random people, which I would say is also quite a good opportunity to just learn to know other people and what they are doing. And over dinner, I was speaking to Franz Kruse, and I was asking him what he's doing. He was asking me what I'm doing. And yeah, we had a conversation about Sjogren's disease and about plasma cells. And he said that nobody is really looking on a detailed level for plasma cells in Sjogren's patients. And I kept that in mind, and we also kept in touch later on. And from that, we grow the idea that after I finish my PhD, I could just take my knowledge on plasma cells, on AACs, and apply it to Sjogren's disease. And with that idea, wrote a grant to the German Research Foundation, and I got lucky, and it was funded. That was how the whole project started.
That's a lovely story, and I feel like so important because it's those small interactions at conferences that kind of change the course of people's career direction.
Yes, I mean, everybody says about networking at conferences and it's really true that, if you don't learn to know other people, then you're not opening up new opportunities.
Yeah, and I'm sure, like every scientist, you have experienced times when data took you in unexpected directions or had experiments that didn't give you the results you hoped for or expected. How have you learned to handle those types of twists and turns in science? And what advice do you have for other early career scientists on reframing unexpected results?
I'm not sure if it's always possible to reframe it. Sometimes it's just, unexpected findings, or you expected a difference and it's completely alike the groups you're studying. I'm always a big fan of trying to report it anyway, which can still, it's getting easier, but sometimes it can still be a little bit challenging to report on experiments that doesn't really show differences where they were expected. I tried to do that a little bit with my PhD thesis. That's full of plots that show no difference, very high p-values, very likely to be any changes there. But I found it's important to still report it somehow, and it's also published then as this PhD thesis. That if other people should be interested in that very small topic, they could find it on the internet and don't make the same experiments again that I already did and find also no results, basically. Sometimes if at first it doesn't seem like a difference or it is unexpected, then of course this can also be very challenging, that you are just forced to think about your hypothesis again. Could I have made some small mistakes that might lead to a different interpretation of the results? Or I also formulated hypotheses in the past and I overlooked some details. And when I finally had the data and it didn't came out as expected, you can also, of course, reformulate your hypothesis and think or look at the data with a different point of view. And sometimes that helps, and sometimes it doesn't help. But of course, it's also important that if you change your hypothesis, that you also then back that up with experiments again, that you don't just adjust your hypothesis to the data. That can be very dangerous.
Yeah. And I guess the flip side of that also is it's important to remember that the science evolves, right? So much as you were kind of describing in the Sjogren story of understanding of cell maturity, as our science evolves, we can get more granular. And so sometimes something you thought was one way actually turns out to have a little bit more dimensionality to it.
Yeah, that really fascinates me about science in general, that nothing is really solid, nothing is really 100% proven. You always have to leave some benefit of a doubt that it might be quite different than everybody expects, even all the experts in the field.
Yeah, I feel like that's one of the exciting things about the field, right? So Tobit, I want to thank you so much for sharing your science and your insights into a scientific career. I am certain we're going to continue to hear more from your research group in the years to come.
Yeah, you're very welcome, Vicky.
And thanks today goes to PhD supervisors and other mentors. I think as we grow as scientists, these people are often like the oak barrels in which wine is aged. They impart their flavor on the scientists they train, and the results mature over time. Thank you for your legacy to the future of our field. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow @ACRheum on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.